Hello everybody, so this is the video for chemistry. We're going to go back to chapter 3 in your blue book, drug and supplement development. And we're going to go through this since we didn't get to do a video on the chapter 3, so that you guys have this lecture portion as well. Be sure to get in there, watch this video, review through the chapter 3 PowerPoint again, make sure you're doing all of your reading, your chapter readings, and then complete the daily assignment. And then again, you need to be participating in both discussion boards. So what is a drug? So the FDA's definition of a drug is a substance that is taken into the body or applied to the body for the purpose of altering the body's biochemical function. So there's various types of drugs. We have prescription, over-the-counter, or OTC, supplements, and homeopathics. Prescription drugs are only available to patients with a written prescription from the provider. You have learned what the required elements of a prescription are, and we have gone through that plenty of times, so I'm not going to kind of touch on that too much, just review back through that. Key point to remember is that controlled substance prescriptions must have the prescriber or the physician's DEA number on it before we can process any controlled substance prescription. So medications are used to prevent illnesses. They're used to treat diseases. They're used to diagnose. And those are done through various types. So we have therapeutic agents, prophylactic agents, destructive agents, pharmacodynamic agents, and diagnostic agents. So therapeutic agents target a specific need in the body, such as the need to relieve pain or relieve symptoms. They can combat an illness, such as an infection, and they can be used to reverse a disease process. So, for example, lowering cholesterol. Prophylactic, we use to prevent an illness or disease. Vaccines are the number one prophylactic thing that we use. We also use germicides, and then some antibiotics can be used to prevent recurrent infections. For example, urinary tract infections. If you have patients that are not able to urinate on their own for various reasons and they have to self-catheterize, because we are introducing that catheter tube into the urethra and the bladder every so many hours throughout the day, typically those patients are on a prophylactic antibiotic to prevent any infections from starting. Destructive agents, they kill living elements, such as cells or infectious agents. So anti-neoplastic drugs or cancer drugs, they are designed to damage fast-growing cancer cells, but unfortunately they can also cause damage to the surrounding or healthy cells. Some antibiotics are bactericidal. We've talked about that in our chapter 4 PowerPoint lecture. So the way I make that connection is think about homicidal, bactericidal. Bactericidal, such as agents such as penicillin, are used to kill the bacteria. And then you have radioactive iodine. Pharmacodynamic agents, they alter the body's function in a specific way. So, for example, if you go in for an eye exam, they're going to ask you if you want to dilate your pupils. That way the ophthalmologist can get a better look all the way in the eye, back to the optic nerve. And those eye drops that they use to dilate your pupils are a pharmacodynamic agent, because it's altering your body, or in this case your eye, in a specific way. Insulins are the same thing. They are used to lower blood glucose, and anesthetics are used to cause numbness. Diagnostic agents are used to help diagnose a medical condition. So, for example, dyes or chemicals such as radiopharmaceuticals that can help prescribers track the action of the cells. So if you've ever gone in for a CT scan and you have what they call a contrast CT scan, they use a particular dye that they put into your IV to kind of track and see and make diagnoses based off of that. So the smallpox was the first vaccine. This was huge for everybody in the entire world. It was developed by Dr. Edward Jenner, and it worked by introducing disease-causing microorganisms, either dead or weakened, into the body. These antigens caused the body to fight back, which created antibodies to neutralize the disease. And smallpox was actually the first recorded eradicated disease by using a vaccine. United States does not give smallpox vaccines anymore because it has been eradicated, but some countries still do, and we still give our military personnel who are going to be going overseas that series of smallpox vaccines. So the necessity of cleanliness and sterility. So during the American Civil War, which was from 1861 to 65, was the microorganisms or the bacterial infections from wounds that killed more soldiers than weapons. It also led to or was a result of the lack of knowledge surrounding the need for cleanliness and sterility during surgeries. During that time, they just set up, makeshift tents and use saws to amputate body parts and use those same saws on everybody that was coming through. And so, as you can imagine, there was no penicillin, there was no medication to give them after the surgeries. The surgeries were not done in a sterile environment, and so most of these soldiers died as a result of infection. So, as a result of this, French scientist Louis Pasteur proved that diseases are caused by microorganisms or germs. So that was one thing for many, many centuries, and in many different cultures, people getting sick were thought to be different things: karma, spirit, evil spirits, those type of things. But Louis Pasteur actually made the connection of, no, when someone gets sick, it's because of this particular germ, or it's caused by a microorganism, or things along that line. So during this time, surgeons began to use chemicals to not only sterilize the wounds, but were also used to help improve the cleanliness of the hospitals. English surgeon Joseph Lister also applied Pasteur's ideas and began soaking surgical instruments in these cleansers, and developed aseptic technique protocols, including hand washing. So think about how long hand washing and aseptic technique protocols and procedures have been in place. It's been in place for a very long time. Obviously, as things have progressed and technology has increased, these things are done in different ways, but the foundation is still pretty much the same. Okay, so diseases carried by insects and other animals. So in 1897, Ronald Ross showed that mosquitoes transmitted infectious diseases such as malaria. Mosquito netting became a huge tool for prevention. They still use mosquito netting, especially in parts of the world that have mosquitoes that transmit malaria. They use drugs like quinine to treat malaria. In fact, one of the thought medications that they're actually trying, the last I heard, is it's kind of being used in New York right now, because New York's in a lot of trouble with the amount of cases of coronavirus that they have. They're kind of the hot spot, and the FDA controlled study of using hydroxychloroquine, which is another drug used to treat malaria, being effective to help these patients recover from the coronavirus. Rodents can transmit the bubonic plague. So during the time of the bubonic plague, the bubonic plague is a bacteria. We still see that same bacteria today. In fact, there was a recent case of a bubonic plague patient, I believe in Wyoming, here I believe was 2019, but they're able to be treated with antibiotics now. When this was going on back during that time, there was no treatment for it, and they didn't understand that the bacteria was spread from fleas. And then you have deer ticks that carry Lyme disease. So antibiotic drug development, a German physician, Robert Koch, isolated microbes that cause anthrax, tuberculosis, and cholera back in the 1880s and the 1890s. So penicillin was our first antibiotic. It was discovered by Dr. Alexander Fleming, and this was a huge victory for the healthcare world, and especially during our time of World War II, because that's when it was first used, and the amount of soldiers' lives saved during that time was tremendous. The scourge of polio. So polio is a disease that results in paralysis of the lower extremities. In 1953, Dr. Jonas Salk was actually the one to develop the vaccine for it. And by 1994, the United States had eradicated completely out polio from our area. We still vaccinate against it. You guys know how I feel about vaccines and why they're important. And this is one of the reasons why we have been able to eradicate diseases that kill children, that kill adults, and they are 100% preventable. So, combating viruses. Okay, this is kind of very, unfortunately, appropriate for what we're going through during this time right now. But the best thing to do here is understand the differences between a virus and a bacteria. So viruses are smaller than the bacteria, and they are a little bit more difficult to treat or combat. HIV emerged in the early 80s. You are watching a documentary on that, and it's a fantastic documentary. It is a little bit of a tearjerker. So not only does it kind of go into the history of how HIV was kind of first discovered in the United States, how long has it been there, and also kind of the effects of a new unknown virus and how we dealt with it in the 80s. And I think it's perfect to show this, not only because we're in this section and we cover HIV, but it's also something important to compare to the response of the United States and the World Health Organization and everything back then, during that time in the early 80s, compared to what our response is now for the coronavirus. The first HIV drug was actually approved in 1987. HIV used to be fatal. In fact, the first cases that they were seeing before the HIV virus was actually even identified, these patients were already in the AIDS stage. And in the AIDS stage, that's the final stage of HIV. That's where patients will eventually succumb to illnesses caused by opportunistic bacteria. So the HIV essentially weakens or totally destroys our immune system, and then it's things like cystic pneumonia or things like that are causing HIV and AIDS patients to pass away because our body can't fight it off. And that's how they first identified the HIV virus. They were seeing very young adult males in the beginning, 21, 22, 23 years old, that should have been healthy, and they were dying from diseases that typically only older people at that time were dying from, particularly the pneumonia. So that's what was raising kind of the red flags early on. Now our HIV patients actually manage the disease by taking a combination of antiviral or antiretroviral medications. But it's important to remember that these medications are not curative. So if the patient stops taking their HIV medications, the HIV infection will take over again. Key discoveries in hormones and hormonal drug treatment. So hormones stimulate tissues or cells of the body to act in certain ways. So some examples of that are estrogen, testosterone, and even insulin. Diabetes, a lot of you covered this in some of your previous assignments. I think this was one of the best discoveries that we've had early on, besides penicillin and vaccines, was diabetic patients prior to the discovery of insulin. It was basically a death sentence. It was only a matter of time because these patients would end up in diabetic comas, and then they would shortly pass away after that. So 2 Canadians, Frederick Banting and Charles Best, were the ones to first isolate insulin in the 1920s, and because of that, we're able to extend the lives of our diabetic patients. Many patients live with diabetes for years, and are able to manage and have a good quality of life. The pill is a name for the birth control. Was first approved in 1960, and still to this day used, and has about a 99% efficacy in preventing pregnancy. So drugs for mental health and cognition. So drug treatment transformed and improved the overall treatment of our mental illnesses and our mentally ill patients. So in 1954, the first mental health drug was approved, and it was Thorazine was actually approved to treat schizophrenia. The antidepressant Prozac was approved in 1987, and then as we approached into the second half of the 20th century, we got things like Adderall and Ritalin. So prior to these medications being available, and more research and all of that done into the mental health aspect, many of our patients were just kind of locked away in asylums or institutions because we, as a society and a medical community, didn't know really how else to deal with them. The Adderall and Ritalin, these are components of amphetamine, the legal amphetamine, and because of that, they are a C2 substance. Anti-neoplastic drugs for cancer treatment, so toxic drugs that actually evolve from weapons of war. The cancer cells grow faster than other cells, and absorb nutrients and toxins faster than other cells. So that's one of the reasons why the chemotherapy is designed and to work, because these cancer cells are kind of pushing out the ability of our healthy cells from getting all of the nutrients and things they need, because they just kind of hog it all. And when we use chemotherapy, they soak that up as well. Cancer drugs are designed to target the fast-growing cells. Unfortunately, most cancer drugs do have toxic effects on the surrounding cells, which can cause harsh side effects. So a lot of our chemotherapy patients have a lot of side effects associated with their treatments. But now there's a lot of research being done to where new cancer medication and radiation treatments are being developed to better target this cancer cells specifically, and help reduce the toxicity to our healthy cells and eliminate or at least reduce some of those side effects associated. So unraveling human genetics. So the gene is a distinct segment of DNA. So in 1953, Crick and Watson developed a model for genetic code, which is the double helix for DNA or of DNA. Specific genes affect specific traits. So whether you have blue eyes, whether you have brown hair, whether you have red hair, whether you have green eyes, all of those things are affected by genes. They also believe in this, when they were first discovering this, that some certain genetic defects cause illnesses, such as hemophilia. Okay, we know Down syndrome is an extra chromosome. So there's a lot of different genetic disorders that are directly related to genes. Research in this field has led to clotting factors for hemophilia and cancer drugs. So if you can hear the dog, I apologize for that. He's obviously not happy. So one of the things that they cover in this HIV video is the almost 100% rate of hemophiliac patients of all ages contracting HIV, and that's because during the time when this all started, the blood banks did not test blood. For HIV, we didn't know what it was. And even they said even if we had been testing, that the HIV virus was so small, it would have gotten through the filtering process anyway, because, we didn't know about this virus and the virus was so small. So either way, so that's how almost 100% of the hemophiliac patients got HIV, from children very young through adults very old who were relying on blood transfusions to treat their hemophilia. And you'll see some of how these people were treated, which is really, really awful. We were a terrible society, and how we treated our HIV patients in the early 80s and even into the late 80s and early 90s. So drugs are being developed that are designed for patients with certain genetic traits. So they're looking into being able to develop specific drugs for specific genes that are a higher risk for specific cancers. So active and inert ingredients. So the difference between an active ingredient and an inert ingredient is as simple as the active ingredient is the biochemically active component of the drug, and the inert is basically the things we put in it to make it a tablet or make it a capsule. We use it to preserve the drug, or it's the vehicle that the active ingredient needs to be in. So for example, a cream, we've mixed the powder of the active ingredient into the cream. The inert ingredient is the cream. Natural sources. So a lot of things can be made or taken from single-celled organisms, plants, animals, minerals, humans. And a couple examples are those of digitalis, was derived from the foxglove plant. Opium, obviously, from the poppy plant, and Taxol was derived from the Pacific yew tree. Synthetic drugs are created from a series of chemical reactions, meaning they do not exist in nature. So an example of that is phenobarbital. So phenobarbital is a synthetic drug. We have created it from a series of chemical reactions, and did not, this particular drug, because it was developed from a series of chemical reactions, does not occur naturally in the environment. Synthetic drugs are created artificially in the lab, but in imitation of a naturally occurring source. So for example, aspirin is synthesized. It mimics the actions of the willow bark tree. And epinephrine, or your EpiPen, we have is used to mimic the adrenaline hormone. So we use EpiPens for anaphylactic allergy reactions, and also we use the EpiPen during codes or cardiac arrest to help to restart the heart. Semi-synthetic drugs, natural drugs that have been chemically modified to either improve the efficacy of a natural product, reduce its side effects, overcome bacterial resistance, or broaden the spectrum of bacteria that can be treated. So for example, amoxicillin, even though amoxicillin is part of the penicillin family, it is specifically a semi-synthetic drug. Biogenetically engineered, so it combines the sciences of biology, chemistry, immunology, and genetic engineering to produce a drug. So for example, synthesized insulins for diabetes, clotting factors for hemophilia, and certain anti-inflammatory drugs used to treat rheumatoid arthritis. So the prescription drug approval process. So some of you got some of these questions wrong. So the FDA is ensuring that approved drugs are proven both safe and effective, okay? Not only do they oversee the safety of the drugs, but also the efficacy. So if they say a new drug comes out and is used to treat allergies, that it proves that is effective in treating allergies. Drugs will undergo a lengthy research process before they hit the market. Strict regulations developed in response to the thalidomide tragedy in the 1960s. Those of you that are going to be coming into law soon, you're going to see that. Those of you that have already had law, you should know what that thalidomide tragedy is. Thalidomide was actually a sedative that was marketed for morning sickness. It had never been tested for safety for pregnant women or babies. And it was determined that it caused some pretty serious birth defects that particularly affected the limbs, arms and legs of the babies. The event catalyzed a major push for safety and efficacy. So each new drug goes through 3 separate phases of research studies. So before starting the studies on humans, the manufacturer must submit an investigational new drug application, an INDA, making the case for human need for the drug and describing the plan for testing. So even before a drug manufacturer can apply for a new drug application, they're applying for an investigational new drug application. And that's where they have to prove that there's a human need and that they are doing this research and they are finding something to help with that human need. So the preclinical research is the synthesis and purification. You have animal testing and the development of the institutional review board. In the human clinical studies, the INDA is submitted, and phase 1, 2, and 3 of those clinical studies is done. Once that is completed, an NDA is submitted, so a new drug application is submitted. The FDA reviews all of that, and at that time we'll make the determination on whether it is approved or not. So the Institutional Review Board, these are typically at universities or hospitals that are attached to universities, because this is where most of the research or human clinical trials are taking place. So this particular board reviews, approves, and monitors medical research that involves humans within the healthcare organization. So the Institutional Review Board, or the IRB, must approve, all of this before the manufacturer can begin the human clinical trials. Part of that requirement is that each research patient must sign and fill out an informed consent. So we've talked about what the informed consent is. The informed consent states the purpose of the clinical trial, what they're going to be taking and all of that, but it also has to list the risks. Since it is a human clinical trial, they cannot ensure that someone is not going to have serious side effects, serious damage done, or death associated with the clinical drug. So after the 3 phases of clinical trials are complete, the manufacturer will then submit the NDA, or the new drug application, to the FDA. The FDA will then evaluate the safety and efficacy of the data all the way through. They will look at the data from beginning to end, and then they will decide ultimately if they're going to approve the drug or not. The FDA may request further studies to be completed, and it may require additional safety labeling. So let's say that they approve the drug, but they noticed a certain patient population had a adverse effect, but it was a small population, maybe a population that had a certain medical condition. They're still going to approve it because the overall benefit to the majority of the American people, or the population, is a good one. So they will require that additional safety labeling to specifically address that small percentage of patients that shouldn't take the drug or should use extra precautions. The FDA may grant accelerated approval to drugs that serve a particular need. We are in the thick of this right now with the COVID-19. They are fast-tracking vaccines. They are fast-tracking treatments. So the use of hydroxychloroquine, and also they have been looking at azithromycin to treat the pneumonia portion of the virus. Both of those do not have an indication specific to the COVID-19, so it's, that's considered off-label use. So they FDA is granting, accelerated approval to get these tried out. And again, I believe New York is the only one in this instance that's doing that. Other countries have. In fact, I think it was France and Italy that the information came to us about these two particular drugs. So, the FDA still has to make sure that it's safe, and is it really going to be effective? So there is some hope, there is some light at the end of the tunnel, but again, it still takes time to gather that data. We're going to go ahead and pause there. So make sure that you are completing your readings. You're going through the chapter PowerPoints again. You're reviewing all of these lecture videos. You're completing your daily assignments, and you're participating each day in the discussion board. If you have any questions, be sure to email me. And other than that, I hope you guys have a good rest of the day.